Okay, let's talk about lipid transport and the diseases. So, let me start out by just telling something about the B proteins. These are apolipoproteins. So, lipoproteins. Now, there are two B proteins. There's B48 and B100. 48 is formed in the intestine, 100 is found in the liver. So 100 liver, 48 intestine. That's how I remember it if you want to. Now, 48 is found in the intestines and it is present in or it goes to the chylomicrons and then obviously the remnant of the chylomicrons. And B100 is formed in the liver and it is present in VLDL, and then of course its remnant is LDL, and then sorry, which is IDL, and then LDL is formed from IDL, so obviously it's also there in LDL, and it's also present in lipoprotein A. Now you have apolipoprotein A1 also, which is part of HDL. Okay, now let's start our story. When you eat food, you will have triglycerides in the food, the dietary triglycerides. You'll also eat cholesterol and minor form of cholesterol esters. Now, in the intestine, you have new de novo B48 synthesis. Now, all of these are absorbed and released into the thoracic duct and then, into the bloodstream in chylomicrons. They are packed in chylomicrons. So, what do the chylomicrons have? B48. From where do they get the B48? From the intestine, newly synthesized, and they have triglycerides. Now, it's now in the bloodstream. We're all happy. Now, in the bloodstream, there's a lot of HDL. Now, what is HDL? It's the good cholesterol. It's the good cholesterol. Now, why am I, particularly telling about cholesterol? Because it wants a lot of cholesterol. So, what does HDL do? It tries to take cholesterol from everywhere. That is one of its functions. Another function of it is that it has these two things called ApoC2 and ApoE. These are formed in the liver, and all the new HDLs will have these two things. Now, mind you, HDL is formed in the liver as well as in the intestine, but these two are formed only in the liver. It's good if you remember that. And all this HDL is constantly being secreted in the bloodstream. It gets secreted. So, or what does the HDL want to do? It's good cholesterol, but it's not really cholesterol. It's something that wants to take up a lot of cholesterol from everywhere in the body. It just runs through the body. Wherever it sees cholesterol, it will pick up the cholesterol, free cholesterol from the plasma membranes of the tissues, even from the vessel macrophages. It's a good thing. Now, these two receptors are there, A1 and then G1. A1 is for, taking up the cholesterol into like a very new nascent HDL. G1 into a mature one. Okay, good. Now, the HDL has something called LCAT. Now, it takes free cholesterol from everywhere in the body, and so that it, gets a lot of free cholesterol, it converts the cholesterols into cholesterol esters, which will move the cholesterol into the core of the HDL. If this is the HDL, like it will move them to the core so that it can get more and more cholesterol. So, it's more efficient. How? Because of the LCAT enzyme. Okay, now where were we? Yeah. The chylomicron with the B48 has entered the bloodstream. Now, in the bloodstream, what was there? A lot of HDL just floating around. What does the HDL have? C2 and E. It will give the C2 and E to the chylomicrons. So now, see, I have written. Now the chylomicrons have B48, E, and 2. And obviously the triglycerides. Now, the chylomicrons, they're flowing in the blood, obviously, like, they came to the bloodstream, they took HDL, lipoproteins from the bloodstream. What will they also encounter while they're flowing in the bloodstream? Adipocytes. There, the adipocytes will have something called LPL, lipoprotein lipase on this substance on their surfaces. Now, the C2 that the chylomicron took from the HDL, that C2 is required so that the adipocyte uses its LPL. So the C2 and LPL are related. Now, what did the chylomicron have? It had the triglycerides, right? So what will LPL is lipoprotein lipase. It's a lipase. It will break down the triglycerides and it will accumulate free fatty acids into the adipocytes. Yay. Okay, good. Now we form a chylomicron remnant. The chylomicron remnant has 48E. It also has C2, but I just removed because its function is over. That's something I will be doing in the video. Now, with the help of that ApoE that it took from the HDL, now the chylomicron will obviously have some cholesterols, whatnot. And if you want a good chylomicron, you want it to be empty. You don't want it to be full. So that's why the LPL is a good thing. C2 is a good thing. Now, the E that HDL gave will help the remnant of the chylomicron because the triglycerides are gone to get inside the liver so that it can be broken down, recycled, and whatever. Good. So, what the chylomicrons do? Triglycerides and cholesterol bloodstream. It took something from the HDL. What did the HDL do? It made the chylomicron competent enough to give the triglycerides that it bought to where it's supposed to give them to adipocytes. So this was the function of the chylomicrons. So where did they start? In the intestine. Where do they end? In the liver. Good. This was chylomicrons. We talked a little about HDL also. Now we have to talk about VLDL, which later on forms IDL, which forms LDL, and then the LDL is what's related to everywhere in the body. HDL was taking cholesterol from everywhere in the body. LDL will give cholesterol to places in the body. So HDL and LDL have inverse functions, opposite functions. So LDL is also called a bad cholesterol. Why? Because it is giving the cholesterol to every place in the body. HDL is a good cholesterol because it takes cholesterol from everywhere in the body. Now, by everywhere in the body, you have all the organs. The organs should not have fat on them. You have the vessels, very important. The vessels have LDL receptors, which means that the LDL can give cholesterol to the vessels. What is cholesterol in the vessels causing? Coronary artery disease, cerebrovascular diseases, all these things which are bad. So, if there's a lot of LDL, it's a bad cholesterol. Why? Because LDL gives cholesterol to the peripheral tissue. If there's a lot of HDL, good cholesterol. Why? Because it is taking cholesterol away from the things or peripheral tissues, vessels or not. Okay, now let's come to the VLDL story. Now, the liver is the place where cholesterol is synthesized. Cholesterol is obviously needs to be sent to the body. Now, liver is where all of the, what's it called? Regulation, if you want to call it, of cholesterol levels are all happening. Why? Because all the dietary is coming up, but if there's too much dietary cholesterol, the liver will do something so that only a required amount of cholesterol is sent to the body. You can't be sending too much cholesterol also. So liver plays a role. If it's bad, then if you eat too much, you exceed the liver's capacity for like a long, long, long time. If you have a very, very, very bad diet for a long time, then only you get coronary artery disease. It does not mean that a perfectly healthy yoga mom decides to eat like a cheeseburger after 25 years and then suddenly she gets atherosclerotic plaques. No, it takes time and it takes a lot of abuse. Okay, where were we? Yeah. So the newly synthesized cholesterol, the triglycerides, and B100, as I said, B100 formed in the liver. All of these are secreted as VLDL. The VLDL has cholesterol and triglycerides. Now, here I wanted to focus a little. The VLDL has triglycerides. Now, comes HDL again with its C2 and E. It will give C2 and E to the VLDL. Why? Because the newly synthesized cholesterol, the newly synthesized triglycerides have to, also go to the adipocyte so that the free fatty acids go inside. So it needs the C2. Why does it need the E, you ask? Because now you might ask, okay, fine. The VLDL was just secreted from the hepatocyte. Why do you want it to come back? Because E was used for coming back to the liver. Now if you can ask what, like, they just wanted to send it out. Why does it want to come back in again? Does something happen in between? Yes, something happens in between that I will come to. Now HDL gives its 2 and E. The 2 will be used by the LPL to, break up the triglycerides and give it to the adipocytes and, make free fatty acids or whatever. Now, HDL had LCAT and it also has CETP, cholesterol ester transfer protein. What does it do? It had cholesterol here, right? This has cholesterol. Now, this HDL will exchange the triglycerides that it has for the cholesterol of the VLDL. So, what is HDL doing? It is accumulating the cholesterol. So, whatever cholesterol was sent by the liver and whatever cholesterol the HDL picked up from the body, it is trying to accumulate in itself. Good. Now, the HDL, good cholesterol, because it wants all the cholesterol, khatam. It is exchanging it with triglycerides. Okay, the triglycerides will go here, blah, blah, blah. Now, something has happened. Now it's a new thing. Now it's called IDL. What will IDL do? It will use the E. It will send back whatever triglycerides it had to the liver back. The triglycerides which the HDL gave from, picking out in the body, it will give it to the VLDL so that the VLDL through IDL can get it back to the liver and make the free fatty acids and, control the metabolism of fatty acids. Now, we had IDL. Now, after this hepatic lipase action, it is now called LDL, where the triglycerides are very low, blah, blah, blah. Now, the LDL, with some lipids, it will go, the peripheral cells have LDL receptors, blah, blah, blah. I don't know, there the LDL is gone. Now, you might ask, okay, why do we even mention this classically? Because macrophages, in vessels have LDL receptor. If there is too much triglycerides that a normal amount of HDL that a normal liver could not handle, and there was a lot of LDL and a lot of triglycerides here, then this increased triglycerides would go into the organs where the LDL receptor is present, and it would cause problems like MI, CAD, CVD, whatever. So that is why this is also talked about. Now, did we talk about everything? Let's check. Chylomicrons, fine. We know how and where chylomicrons go. VLDL, it becomes IDL, which becomes HDL, and HDL goes away. What is one thing that we are forgetting? HDL. What happens to the HDL, which is, accumulated all the cholesterol? There is a receptor called SRB1, which will help the HDL go back to the liver and dump all the cholesterol. So, the newly synthesized cholesterol or the recycled cholesterol, whatever, the HDL will come taken back to the liver. So, from everywhere in the body, all of that cholesterol will go to HDL. Even the dietary cholesterol, you know, everything will try to go to the HDL, which will take it back to the liver from the body. Now, HDL was a scavenger. The HDL is helping in so many ways. How is it helping? It is giving the triglycerides to the VLDL so that it can do its thing, so that It's the triglycerides here, they were taken up by the adipocytes. Only by the adipocytes, and very few here. But even the liver wants some fatty acids to, synthesize some things or send it to other places. It also wants some fatty acids. Where will they come from? The HDL, which gives the triglycerides to the VLDL. And the HDL also has a important function here. It makes all the things functional. All this adipocyte thing and all, the liver, handling all these things are all function because of HDL. So this was the physiology here. Now let's just, fastly discuss the diseases. This A beta lipoproteinemia. So A means none, like it's not there. Then B lipoproteins. The B proteins are not there. So 48 is not there, 100 is not there. So which means that chylomicrons are not there, VLDL, LDL, IDL, nothing is there. So many functions does not happen. So if there's no chylomicrons, if this is not there, then all of this will accumulate in the lumen of the intestine, which will cause steatorrhea, abdominal distension, and fat malabsorption will cause what? The fat soluble vitamins will also not come. So vitamin A is important, E is important. ATAX and others are there. You can just go and read. Now the familial dyslipidemias, there are actually five, but five is basically just like one and four combined. So you have, basically four and four also you have A and B. You know, B is just this plus VLDL, I think. So you can, read that. Now one is hyperchylomicronemia. That's how I remember it. That one is, related to chylomicron. These are all hyperlipidemias. So, hyper, whatever. Now, okay, this pattern, autosomal recessive, you remember this ARAD, ARAD, you know, just have to remember. Now, hyperchylomicronemia happens because of an LPL deficiency or sometimes C2. LPL and C2 are related. So, if there's no lipoprotein lipase, which means that the adipocyte taking up will not be there, all the triglycerides will not be given to places, which means that the triglycerides will increase, and increased triglycerides are related to recurrent pancreatitis. Cholesterol also will increase because all the cholesterol is also there, obviously. But this also you have to. This is the main thing that I wanted to remember. Now, hypercholesterolemia is because of LDL receptor. Now, say there is a problem with, now what is LDL having? All the triglycerides are gone, so it has cholesterol. The LDL has cholesterol, which it gives. If there's too much cholesterol, it will give the too much cholesterol to peripheral cell, which will cause the plaques and all, cholesterol plaques, whatever. So this LDL receptor is not there, so the cholesterol things cannot happen. So the LDL which has cholesterol, so increase cholesterol. Now, 3 is E, so there's no ApoE, so there's no this chylomicron is involved, there's no this is involved. So chylomicron and VLDL, and VLDL related to triglycerides, chylomicron related to triglycerides, all this lipid here. So increase triglycerides. Now, 4 is VLDL, has 4 letters, so overproduction of VLDL. So if there's too much VLDL, what will happen? There will be too many triglycerides. Yes, VLDL is related to triglycerides. If there's too much VLDL, the HDL will give too much cholesterol, too much, triglycerides, blah blah whatever, and then this will happen.